Uh, Thank you for inviting me and giving me the opportunity to present uh, some basic immunology titles. Um, uh, but actually, it's a very wide thing, and there there isn't quite a distinct link between one to the other. So there are somewhat disconnected, but we will go over, and I would like to make it as simple as possible. Uh, I will talk first about the complement system, which is similar to the coagulation system of protein. As you know that there are several proteins which one activate the other, then the other activate the other, like a cascade uh, to to cause clotting. The complement system facilitate the immune function of the body to eliminate um, microorganisms, tumor antigen or self antigen. There are more than 30 proteins or glycoproteins which constitute the complement system. Uh, the Some of them which take part in active complement system, they are numbered as C1, 2, 3, 4, 5, 6, 7, 8, 9. As you see, the numbering 4 comes before 2. This is how they are sequentially activated. One would have thought that it would have been better if C1, C2, C3, C4, and gets activated in that way. But here there is a bit of kind of a different thing. 4 comes before 2 in the activation pathway. The 5, 6, 7, 8, 9 complement components, they are called as the final pathway. And these C1 to C3, they plays in the initial part. The initial pathway of activation is mediated by 3 ways. They are called classical, alternative, or lactin. But the final pathways are common to whichever pathway has been activated. The alternative system is phylogenetically more primitive. It's much older than the classical pathway, and it's operational in snails, very primitive animals, and the classical pathway came into being in subsequently. Each component activation leads to 2 products, one large and a small one, usually called A and B. But there are 2 exceptions to this rule also. There are 3 pathways of complement activation: the classical, the alternative, and the lectin pathway. This cartoon from a textbook of Abbas and others, I took this cartoon because it's quite good, you know. You see this molecule, C1, as a pentameric molecule. And we let's imagine this should be a bacterial cell, or it could be a red cell in the process of if any antibody is targeted against cell structures, you can just visualize this. And what happens, these complements are soluble products. They are circulating in the serum. But upon some signal, the C1, which is a big molecule, which is a pentamolecule, it binds to antibody, and there are specific reasons in the antibody. You've heard about the CH2 domain of IgG and CH3 domain of IgM. That is the site where complement binds. So if an antibody binds to a bacteria, the complement anchors onto it, the C1q. And once C1q anchors onto it, then the C4 get attached. It's inactive form, it gets activated. But then there is cleavage of C4 by an enzyme called C1R2S2. These are part of the C1 system, and the C4A is liberated and C4B gets attached. And so binding of C2 to C4, that is the next step. That is crucial because that causes formation of C3 convertase. And C3 convertase is a pivotal enzyme for all the three pathways. And the C3 convertase in the classical pathway is called C4B and 2A. It's a complex thing. And then the C3 convertase cleaves this molecule, the C3, into C3A and C3B. The A is a small part, it goes away. The C3B joins with C4B, C2A, and you can see a molecular association on the bacterial cell. Means they are sitting like one member joining the other member gets activated, and then this complex C3B, C4B, C2A, that forms the C5A convertase. So, and all is happening on a cell membrane. You can imagine on a bacteria like pneumococcus. So the antibody sit on the bacteria, or you can see in an autoreactive situation, it can be on, say, a beta cell in ITDM case, and that engages these small proteins. But they are very powerful biologically. We will come to that. Here we will little go back to find out what are the proteins in the classical pathway, the early activation. C1q is a big protein of 750 kilodalton that initiate. C1q is a hexamer of 3 pairs of chains, C1R, C1S. C4 comes next in the sequence, and then C2. And the biological function of this C1q, it binds to the Fc portion of antibody that has bound to antigen or apoptotic cell. C1R is a serine protease, it cleaves C1S. C1S is again a serine protease which cleaves C4 and C2. C4B covalently binds to the surface of microbial cell where antibody is bound and complement is activated. C2A is a serine protease, means all are enzymes, and that cleaves on the subsequent. And you've got to remember that C3 convertase is the central anchorage enzyme that goes into the late activation pathway. The alternative pathway is very much part of the innate immune system, as Dr. Bhaskar Shah was alluding to adaptive and innate. This is an innate immune system pathway, facilitatory protein system. This and the initiation is independent of antibody antigen interaction. You don't need an antibody antigen interaction, and it can be initiated by 3 ways. And first is it's a takeover pathway. C3 molecules slowly get proteolysis, and C3b and C3a are liberated. And once C3b activates, get joined with factor B and D and properdin, it makes C3 convertase. It can be initiated by a protein called properdin or by proteases like thrombin and kallikrein. So, as I told you, the C3 undergoes spontaneous hydrolysis, and it causes inactivation of C3b in the fluid stage. And this C3b, it binds to microbial surface and binds to factor B. And cleavage of factor B by factor D and stabilization by properdin forms the C3b and BB. BB, the other B stands is a small b. That is the C3 convertase. And this cleaves many molecules of C3 into C3b and C3a. And you have got an amplified or many molecules of C3b, which can bind to C3b on microbial surface and engage BB to form, to add on C5 to form C5 convertase. So this is independent of antigen antibody interaction. It can get activated like this. So the main proteins are C3, factor B, and factor D, and properdin. And we talk about factor B. BB is a serine protease that active enzymes of the C3 and C5 convertase. And factor D is a plasma serine protease that cleaves factor B, C3b, and properdin stabilizes C3 convertase. So here C3 convertase is C3BB, factor BBB. But for the classical pathway, this is the C3 convertase, the C4b and 2b. This is the lectin pathway. Initiated when small proteins recognize microbial. Lectins recognize specific carbohydrate component primarily found on microbial surface. And again, this is part of the innate system. And there are various proteins of the lectin pathway. So this is also independent of antigen antibody complexes. And basically, these proteins are mannose binding lectin, M-ficolin, L-ficolin, H-ficolin, and they can also lead to complement activation. Now, once this thing forms, the C3b, BB, and C3b, that is from the alternate pathway, C5 gets activated, and C5a, C5 gets split or digested to form 2 molecules. One is C5a and C5b. This C5a is one of the most powerful anaphylar toxin in the biological system. It's tremendously pro-inflammatory. So that causes that causes inflammation or intensify inflammation. And the other molecule C5b rest on the C3b, BB, and C3b. So gradually the complex is getting formed. And to this complex, the terminal pathway complement C6, C7, C8, they simply add on. They don't need kind of an activation, a liberation of a small molecule. And when they add on to this, the C9, this is called as a membrane attack complex. And you can see how C9 molecule is like a tunnel. And this terminally membrane attack complex, this forms a tunnel in the lipid membrane or the plasma membrane of the microbes or the cell. And once that hole is prepared, and so the cell undergo lysis. I mean, the so actually it punches hole, and there are beautiful pictures you can see in patients with autoimmune hemolytic anemia. You can see red cells. If you do an electron microgram photograph, there are holes on it, and you can see those pictures in Google Images. And even microbes such as pneumococcal pneumonia, how if antibody is generated, the cells are killed. Once a hole is there, you would know that the osmolar concentration would be different between the inside of the cell and outside of the cell, leading to. So the proteins of the late stage of complement activation, what are these? C5, C6, C7, C8, and C9. This C5b is the initiate assembly. And you just have to remember that C5a is a very powerful anaphylatoxin. That is there, it causes accumulation of more inflammatory immune mediated cells into the site of reaction. So this is a component of MAC C6, it binds to C5b and accepts C7. C7 is a component of the MAC bind to C5b and 6 and inserts into the lipid membrane. And you can see the beautiful way the cartoon has been made, how holes are being formed on the cell surface. So this is a membrane attack complex. C5b gets to C6, C6 is attached to it, and C8 and C9. And then we have a cylinder of C9, which causes holes on the cell surface. So there are, for this complement, there are complement receptor. We have C1 inhibitor, factor I, factor H. The significance of it will touch in a minute. C4 binding protein, membrane cofactor for protein, decay accelerating factor, CD59, and so on. And this C1 inhibitor, it's a serine protease inhibitor. It binds to C1R, S, and dissociate them from C1Q. So if this inhibitor is absent, C1 inhibitor is absent, there would be more of classical pathway activation. And factor I cause a serine protease, it cleaves C3b and C4b but using factor H. And these are regulatory for the alternative pathways. So C4 binding protein, it binds to C4b and displaces C2. So this is again a checkpoint inhibitor. So what does the complement do? The complement, the C3b, binding of C3b to microbes has a good opsonizing potential. So the microbes like the phagocyte like to eat up the the phagocytic cell like to eat up microbes which are coated with C3b. I mean, it's something like a butter on a toast. I mean, you'd like to eat uh, butter which is layered over a toast, and similarly C3b, if it is on the microbe, so it makes it palatable for the phagocytic cell. It stimulates inflammatory reaction. I told you that C5a and C5a particularly is a very powerful anaphylactic toxin, and it can recruit and activation of leukocyte by both C5a and C3a. C5a, recently there is a drug that has come which can inhibit C5a and has been proven to be a drug in ANCA-associated vasculitis, and I mean, a lot of excitement is about this drug that you can manage even without steroid. So it is, you would have guessed the drug name by now. And then it causes complement-mediated cytolysis. As I told you, that it punctures a hole in bacteria or red cell, causing osmotic lysis of the bacteria. So binds and opsonize bacteria and make them susceptible to receptor-mediated phagocyte. It elicits inflammatory response. It interferes with components of the adaptive enzyme, clear immune complexes from the serum and or eliminate apoptotic cells. And the membrane attack complex directly kills some pathogens by creating pores in the membrane. So this is an overview, whether it's a classical alternative or lectin pathway. The C3b is activated, C3 is activated, and the C3b gets deposited on the microbial surface or on the cell membrane. The C3a is an anaphylactic, is an inflammatory, biologically active inflammatory mediator. It causes recruitment and activation of leukocyte, and the C3b on the surface of the bacteria makes them easily phagocytosed by our cells with phagocyte, the macrophage and the WBC, and it also forms a membrane attack complex causing lysis of microbes. So complement, as the name suggests, is facilitatory to elimination of microbe, foreign tumor cells, or autoreactive. Because it's a powerful system, we need to have checks and control. Otherwise, there will be too much of inflammation and too much of immune-mediated reaction. This C1 inhibitor is one such protein. And you know, those who are deficient in C1 inhibitor have angioneurotic edema because the classical pathway gets activated. Then formation of the C4b 2b complex classical pathway C3 convertase. There's a molecule DAF and CR1 formation complex. DAF, MCP, and CR1 displaces C2b from C4b. So this molecule C4b and C2b, these are the proteins DAF, decay accelerating factor, MCP, and C1. It dissociates, so further engagement doesn't occur. But if you have deficiency of these proteins, then you can have problem, like you can have hemolysis. A condition called paroxysmal nocturnal hemolysis is because of deficiency of DAP protein. Formation of C3BB complex alternative pathway C3 convertase DAF and CR1 displaces BB from C3b. So this is the checkpoint or regulates the activation of both classical and alternative pathway. C3b, MCP and CR1 acts as cofactor for factor I mediated proteolytic cleavage. Factor I can do proteolytic cleavage of this complex, C3b or C4b. When it gets, factor I can displace that. So in absence of this factor I, you can get a cell lysis. Then we have activation of the late activation, a molecule called CD59 inhibits the poly C9 formation, which you saw as a tunnel. CD59 inhibits that. So even in the late assembly of the complement pathway can be inhibited by this molecule. And S protein inhibits membrane insertion of C5b. So you can have inhibition of MAC formation, inhibition of MAC formation by these two molecules, CD59 and the S protein. There are individuals which are genetically deficient in complement components. If one has C2 deficiency or C4 deficiency, they develop SLE, and usually they are very severe forms, and they almost die in neonatal or infant period. C3 deficiency is unthinkable to sustain life, because that's the final convergent pathway of these. And if you have absence of C3, then you cannot tackle bacterial infections. Again, deficiency in properdin and factor D, you have increased susceptibility to infection with pyogenic bacteria. And if you have deficiency in this terminal complement MAC, you can have Neisseria infection and mannose binding lectin deficiency causes serious pyogenic infection. And deficiency in regulatory protein, if this regulatory protein factor A is deficiency, then there will be depleted plasma C3 with pyogenic bacterial infection. Factor A deficiency causes excessive alternate pathway activation and consumption of C3 and glomerulonephritis. Factor H mutation is atypical hemolytic uremic syndrome, and factor I and factor H combined deficiency will have autoantibody C3 nephritic factor glomerulonephritis. If you have deficiency of the complement receptor CR3 and CO4, again you have recurrent pyogenic infection. The microbes have machinery to evade complement mediated lysis, and they can interfere with antibody complement interaction, bind and inactivate complement proteins, the protease mediated destruction of complement component, and microbes can mimic regulatory component proteins and cause inhibition of the complement activation. In practice, what we generally, clinical practice, what we generally assess is C3 and C4. C3 is for both pathways, C4 is for classical pathway. If C3 and C4 are less, then we see it in usually situation like SLE, and that is taken as an active marker for SLE. But if only C3 is low and C4 is normal, it means alternative pathway activation is there. I think for physician, it is important to bear that estimation of C3, C4 are useful in immune complexes mediated disease, such as SLA or serum sickness. And if the value C3 and C4 values are less, one can postulate that there is active disease, and with proper treatment, the C3 and C4 value normalizes. We do monitor C3 and C4 in patients with SLE, particularly lupus nephritis. And another practical thing, you may have been seeing a lot of angioneurotic edema. We usually do both protein and functional assay of C1q inhibitor. Let's stop there.
Thank you, sir, for your first part. Already our chat box and QA session is flooded over questions. So I have two questions for you only.
Okay.
First question from Dr. Maisum Sobahan. Sir, which complement pathway is more effective and why?
Oh, both are very effective. Both are very effective. Both classical and alternative and the lectin pathway, they are all effective because once C3 gets activated, the final common pathway is same. I couldn't tell. There are, if it involves the adaptive immune system, antigen antibody, it is the classical pathway that is operational. But if antigen antibody is not, immune complexes is not involved, it is the alternative pathway or the MBL pathway. So both are equally effective.
Thank you, sir. Another question from Dr. Mahmudul Hassan. His question is, sir, what are the presentation of complement deficiency? First question, which complement deficiency is common or which one is most dangerous? Basically, there is three questions in one.
He's very good in asking questions.
Yes, sir. Our participants are very good in asking questions.
Yeah, and sometimes questions are difficult to answer. The first question was, if you are given a choice, which
presentation of complement deficiency?
Complement deficiency results in immune complex disease, because complement deficiency result in immune complex disease like SLE or pyogenic infection. That is the answer. C2 and C4 deficiencies, that means deficiency of the early pathways, they can have SLE-like feature in a child or in an infant. They have usually malar rash, the C4 deficiency. C2 deficiency, the disease is so severe that they probably die in utero. And C3 deficiency leads to serious pyogenic infection, and I don't think C3 deficiency they able to sustain survival. Other factor deficiency like properdin factor D, they cause increased susceptibility to infection. That means they have recurrent infection. But I would think, I don't know which one is the common, because deficiency of these early factor and late factor, sometimes they cause a death in utero. So it's very difficult to study. Usually the common thing that we see is C4 deficiency. And that too, C4 has two locus or two alleles. C4, one of the allele deficiency, you can have SLE-like features, but they are generally mild disease. And C4 would be persistently low. We've seen some SLE patients whose C4 are persistently low. We believe that they are deficient in C4.
Sir, shall we continue our question answer session, or you will go your next part, sir?
I think it's up to you.
Okay, sir. One last question. Okay, sir. One last question. Sir, tell me about the immunological findings or immunological development in SLE.
Immunological abnormalities.
Abnormality, or what is the update about immunological condition of SLE? সবগুলো মিউট করেন প্লিজ, কন্ট্রোল থেকে সব মিউট করেন, কন্ট্রোল প্লিজ মিউট। Okay, sir. What about SLE? Last question, sir, for you. Misra sir ko on karen, Misra sir can mute karen. Unmute Mr. Sir, please.
Yeah, can you hear me?
Yes, sir.
Yes, we're hearing, sir.
Yeah, the thing about SLE is that you must be all knowing that it's antibody-mediated immune complex disease. And if you want to know update, the main update is trying to inhibit or inhibit SLE by biological agents. Nice talking with you. One of the thing is, since the B cell produced autoantibodies, one line of treatment is by rituximab, so that B cells are killed, but the plasma cell escape, so long-lived memory cell escape, so the other defense system for against tetanus or other microbes that is persistent. And the second drug that is available for targeted therapy is called belimumab, which is against the BAF protein, which is a stimulator for B cells. So that is something which have, unfortunately, it's not very active in severe cases, but it does help in maintaining. It's a good maintenance drug, providing occurrence of relapse. That is prevented. So, and also the work is going on to inhibit various kinase inhibitor to see that their effect in, because these kinase inhibitor would affect on a broad panel of cytokines. And SLE is generally viewed, it's actually both T cell as well as a B cell mediated disease, such as the nephritis part. There is both antibody is also acting, and the TS17 cells are working. So the kinase inhibitor are a good exploring a good opportunity to block several cytokines. We have also proteasome inhibitor, like those drugs which are given in myeloma. They are being tried so that you can target the plasma cells which are producing antibodies. So that is sort of update that is going on, that to reduce the antibody level, there are cells which produce them, and that is usually the plasma cells or B cells. And in the direction of T cell, we still do not have much success. Mostly the success has come from B cell, targeting B cell and its product.
Okay, sir. Thank you, sir. Now your next part, sir, please.
Yeah. I see Deversist now coming.
Yes, sir. He joined our course coordinator come faculty. Professor Devashi Janda sir joined now. He's here with us.
I am so grateful to Devashish for bringing me in, but sometimes I get mad upon him for giving such an extensive talk. I mean, on a topic which is which encompasses complement, phagocytosis, MHC, and I'm actually a very simpleton. I cannot I cannot do right now. Let's continue with my talk. I told you that complement, it coats the microbe like a bacteria, like a butter. We coat the you just imagine a situation where antibody is binding to a microbe and complement C3b is there. So you have two ways to eliminate that. One is you can punch a hole in the bacteria, and the other one is you make it palatable for the phagocytosis, for its phagocytosis. Phagocytosis is the first layer of defense. I mean, the first layer is, of course, the skin and the intact mucous membrane. But once the microbe enters that, if we have a, if that is, the barrier is broken, then comes these cells, which are the front runner. I mean, and these front runner cells, they don't know anything. They will just take it up like what amoeba does. You will all remember your biology classes in school or intermediate, that what an amoeba does, it just engulfs anything that is floating around it, so that is the food for the amoeba. So the food for the phagocytosis, for the macrophage or the neutrophil, is that it has a dynamic membrane, and once a C3b or antibody or C3b binds on cells like that, it makes them more palatable to just eat it up. Eat me, that is what phagocytosis does, and it swallows them into and makes a vesicle of it called the phagosome, and the phagosome is gradually swollen, and powerful enzymes are there which digest those protein part of the invading microbe. And the microbe is eliminated, and as Dr. Sahab was telling you, and I will be also telling you, that this is a stepway for it to present an antigen to adaptive cells. So the phagocytic cell are part of innate cells, but they provide stimulus to the adaptive cell, which we'll see later. And these are the receptor through which the macrophage makes a synaptic synapse with the fungal, with the microbe. There is dectin-1 protein, and that protein, if it engages with the microbe, it causes not only phagocytosis, but also it engages a cascade of signaling so that pro-inflammatory cytokine are produced. So, and interferon gamma is produced, and nearby cells are kind of activated. It sends a warning signal. So these are the receptor. One of the best receptor for phagocytosis, which are opsonic receptor, is an immunoglobulin molecule. That is, immunoglobulin molecule has two sides. One is called the FC side and one is called the FAB side. The FAB side is for antigen binding. The FC receptor, the macrophages or the phagocytic cells, they have FC receptor on them to attach to the immunoglobulin. And they have complement receptor CR1, CR3, and CR4. And you will remember the C3b receptor. The C3b binds to these receptors. And they also have alpha 5 beta 1 integrin. And there are non-opsonic phagocytosis. They are dectin-1, macrophage receptor Marco, scavenger receptor A, and a toll-like receptor, which you may have heard before. And what are the ligands for this? The FC receptor is for antibody, and the complement receptor is for complement opsonizing target. And we have dectin for beta glucan, and macrophage receptor for bacteria undefined specific ligands. With that, we come to the major histocompatibility complex, which was also referred to in Dr. Saha's talk. The MSC is such a crucial cell surface molecule for antigen recognition. It's a histocompatibility complex linked cluster of genes which products play a role in intracellular recognition between self and non-self. The MSC is a region of multiple loci that play a major role in determining whether transplanted tissue is accepted or rejected, which is histoincompatible. There are two types of MSC molecule, MSC1 and MSC2. They are different. MSC1 has got a single chain of polypeptide, whereas a class 2 molecule have two chain of polypeptide, the alpha chain and the beta chain. The class 1 molecule have got a single chain, and these are the various domain, the globular structure, the alpha 1, alpha 2, and alpha 3. As you can see, the peptides bind between alpha 1 and alpha 2 in the class 1 molecule, whereas in the class 2 molecule, it binds between the alpha 1 and beta 1 domain. Here it is alpha 1 and alpha 2 because there are no two chains. With this chain, there is a beta 2 microglobulin. This is not a polymorphic molecule. This alpha molecule is polymorphic. That means it is different from what is in one individual from the other, whereas the class 2 molecule, of course, is highly polymorphic. Both the alpha 1 and beta 2 molecule, they have hypervariable region or molecule which is different from one to other to actually tackle with a wide variety of antigens which are diverse. This CD8 cells, it binds to alpha 3 domain of the MSC class 1 molecule. Here, as I told you that the class 1 molecule is got a single chain, whereas the class 2 molecule have both alpha and beta chain, and the CD4 binds to the beta 2 domain of MSC class 2 cells. This is important. MSC class 1 binds to the CD8 T cell, and the MSC class 2 molecule binds to the CD4 T cell. This is the structural organization of human and mice. in mice, it is called as H2. Much of our research initially has been on the mice system, but we have almost a similar diversity in human. It's called the HLA. And there are three set of locus, the class 1, the class 3, and class 2. Class 1 has A, B, and C, which is not exactly sequential. We have HLA-A, HLA-C, and HLA-B. And class 2, we have got two locus, DR, DQ, and DP. We have several DR alleles. So we can, you can have DR, DQ, and DP. In human disease, usually we know of the B, the polymorphism of B is associated with, like B27 is associated with spondyloarthropathy or Angst-Pond. But and rheumatoid arthritis is associated with certain locus of DR. And you know, diabetes mellitus is again associated with certain alleles of DQ and DR. Similarly, when we make a genetic association with disease, we try to, when we say that it is B27 or B52 with a particular disease, it means it's the B locus allele, it is associated with that disease. This is the organization in the human chromosome 6 of the class 1 and class 2 genes. And you can see the HLA-G, HLA-A, HLA-E, and in between 1 and 2 are these locus, the locus for TNF and the lymphotoxin alpha. And then we have DR alpha, DR beta, TQ alpha, DQ beta. And because of this alpha and beta chain also, it's a highly variable or polymorphic, that is what term is used, that these molecules are. So we have extreme difficulty to find a match if we are not related within, I mean, you may get a match between brother and sister, but of course a monozygotic twin will match at all locus. When you refer of someone HLA, we call it HLA A4, A6, which means one of the locus has been inherited from the father and the other one is from the mother. If you have HLA4, that means you are homozygous at that site, and both have been both the 4 locus have been inherited from your parents. You are called homozygous. Generally, we are, you know, we have 2 loci so we have HLA 4, 6, then we may have B27, 51, and C some other number, and we have DR, DP, DQ. We generally don't refer to DPU and DQ, but now with molecular typing, you can really find a very complicated HLA. Like your name is unique to you, such as your HLA typing is unique to you. And earlier it was lymphocytotoxicity was done to assay this. Now we have molecular typing before cross match. You know that you have to have at least 4 or 6 locus match before transplant can be possible. Now, I will talk very briefly on antigen presentation. And antigen presentation, Dr. Saha has told all about adaptive immune system. you know that we have a cell-mediated immune response and we have a humoral immune response. All cell-mediated immune response, the macrophage present the antigen of phagocyte macrophage to the effector T cells. T cell cannot see the antigen directly, and monocyte migrate to the site of infection, upregulate the class 2 MHC, and present antigen in the context of the molecule to the T cell. The T cells cannot see antigens directly. The cell has to take up the microbe, digest it, and present it through class 2 or class 1 molecule to the T cell. Class 1 molecule is present to the CD8 T cell, that is the cytotoxic cell, and class 2 molecule is to the helper T cell, CD4. The humoral response, B lymphocyte, it internalize soluble protein, and of course it can present antigen to the T helper cell. That is, B cell also can be an antigen presenting cell. All nucleated cells present class 1 associated peptide derived from cytosolic protein antigen. So we have essentially two pathways of antigen presentation, class 1 and class 2. Protein antigens in class 1, protein antigens present in acidic vesicular compartment are presented on the cell surface. In class 2, the antigens they present in the cytosol. Here it is present in vesicles. If an antigen is taken up by the antigen present in the cell or the macrophage, and you know it is processed in this vesicular complex, and then it is presented to the class 2. That means it is presented by this way. It cannot be present to the class 1 pathway to the CD8 cells. But endogenously, if the antigen is there, it is transfected like an ovalbumin gene is transfected into the cell, then it can be presented by the class 1 cells. So a cell which takes up protein by the process of endocytosis is presented by the class 2 cell, the black one here. But if there are like virus and tumor cells which are there inside the cell, they are presented by the class 1 to the CD8 cell, and this is to the CD4 cell. You can artificially introduce foreign protein antigen into the cytoplasm, and then they can be presented by class 1 cells. But if the cell engulfs this protein, that will be presented by the class 2 cells. So the stage of antigen presentation by class 2 is uptake first, then processing in endosomal and lysosomal vesicles, and class 2 molecules are synthesized in the endoplasmic reticulum and transposed to endosome, and association of class 2 molecule with peptides. And diagrammatically, as we can see, so this is, you just imagine this to be a cell, and a protein antigen is engulfed by the cell, and there are proteases in the lysosome which join with the endosome and digest it. When they are digested, and then at the same time, the class 2 molecules have a molecule called CLIP sitting on it. This CLIP molecule is dislodged by a molecule called DM, so that this molecule, it comes in this exocytic vesicle, the class 2 molecules, which have the clip on it. This is produced in the endoplasmic reticulum. It joins with lysosome, and then this molecule DM, it clips this clip molecule, so that this clip molecule is dislodged, and then the peptide molecule get lodged onto the MHC class 2 molecules, and then it is transported by vesicles to the cell surface for presenting to the CD4 T helper cell. So the class 2 molecules are synthesized here. They are alpha and beta chain. It has a molecule that plugs in initially, and this molecule, and when the lysosome meets with this vesicle here, this is called as antigen processing. And usually, as Dr. Saw was referring, that a certain number of amino acid can only fit in this space. They have to be usually up to 9 or 13 mer peptide. Means the peptide can only have 9 to 13 peptide. He was telling you about how much a class 1 molecule engage and how much is a class 2 molecule. We'll come to that. So it takes some time before the antigen is presented, but you do a glutaraldehyde fixation, then glutaraldehyde basically fixes this process of antigen processing, and then you will not have antigen presentation. But so you can know if it is fixed 3 hours later, you can still present the antigen, but if it is done before 3 hours, then the antigen cannot be processed. This is the same thing in a magnified view to tell you clearly that the class 2 molecules are synthesized in the endoplasmic reticulum, and this class 2 molecule are transported with a chain which is called as invariant chain, and then the protein molecule, there is digestion of this molecule proteolytically is degraded, so that this becomes empty to attach a peptide antigen. This HLA-DM molecule catalyzes the clip molecule, and then peptide molecule are engaged in the endosome, and through a vesicle, the class 2 molecule are transported out for presenting to the CD4 T cells. For processing of cytosolic antigen by class 1 MHC, usually certain antigens I told you, they could be the virus, which are cell inside the cell, like the COVID virus, or tumors, or intracellular bacteria. And these are proteolytically degraded by proteasome and transport the peptide from the cytosol to the endoplasmic reticulum, and the assembly of peptide with class 1 molecules with the peptide and surface expression. So you have the virus here, and these are synthesis of the viral protein. The proteins are ubiquitinated, and they go to, this is a mega enzyme complex. It's called the proteasome. It's a very powerful enzyme complex that degrade all protein molecules which are ubiquitinated. I mean that these proteins, they have a ubiquitinating. All normal protein that are not required by us, antiviral protein or the tumor protein, if it gets, they are digested by the proteosomes into small peptides. And these peptides, through a transport associated protein or TAP protein, it enters the endoplasmic reticulum to get attached to a protein called ERAP, and ERAP further make a minor editing of this peptide, and then this peptide is you need a very fine processing of this peptide to engage into the class 1 molecule. The class 1 molecule is a single chain. There is a small dash here that's a beta 2 microglobulin, and then again, like, it comes into the Golgi apparatus and in the vesicle and present itself to the CD8 cytotoxic T cells. And once this is presented, the cytotoxic T lymphocyte actually kills the entire cell. So the cytotoxic T lymphocyte, if a cell, antigen presenting cell, presents a structure of virus or tumor or anything to the CD8 T cell, it recognizes that this is foreign and it kills it. Through it's got good killing mechanisms, you may have been told. This proteasomal degradation is a large multiprotein enzyme complex. I told you it's a very huge complex protein which normally is required to clean the cells of all unwanted protein. You know, several proteins are synthesized which are not required by the cell, and they are ubiquitinated, and that is the signal so that proteasomes degrade those proteins. So cytosolic protein are degraded by the proteasome into small peptide with transport associated protein. They gain access into the endoplasmic reticulum. It gets into the groove of the MHC, and through Golgi apparatus, it is presented to the CD8. So, I mean, this is a comparative chart of class 2 and class 1 MHC pathway. Seriously. Class 2 molecules are, for example, DR molecule. Class 1 molecule is something like B27. That would easily relate to our rheumatic disease. So, you know, DR4 is associated with rheumatoid arthritis. That's a class 2 molecule. But here we are talking not of association with a disease. Here we are talking what they actually do in the antigen presentation. Class 2 have a stable peptide MHC complex, and class 1, the peptide fits in between alpha 1 beta chain. Here it is only one chain, so it is fitting with the alpha chain only. What are the types of antigen presenting cell? It's the dendritic cell, mononuclear phagocyte, B lymphocyte, endothelial cell, thymic epithelium, all nucleated cells. Remember, class 2 molecules are normally quiet on the cell surface. But if they get a signal for inflammation, they are upregulated. But class 1 is there all the time. It is response to CD4 T cell. This is for CD8 T cell. The source of protein and antigen is endosomal, lysosomal protein, and these are cytosolic proteins. Enzyme responsible are endosomal or lysosomal protease. This is a cytosolic proteasome. And site of peptide loading is specialized vesicular compartment. This is the endoplasmic reticulum. and chaperones in the endoplasmic reticulum or invariant chain, and there are these chaperones and tap. I will stop here because if I go on, I will get very bored. I mean, you will get very bored. This is no doubt very interesting, but we have dealt a lot of topics, and I would prefer to stop here and say hello to Saeed Atiul Haq, whom I see now. It's such a wonderful to have you on the screen.